Hi there, DrBiology here. This is an A-level video and it's related to the Hardy-Weinberg Principle or Hardy-Weinberg Equilibrium. I've named it a Masterclass because I'm going to go through the basics of the principle, tell you a bit about the history of it, and then we're going to go through some examples. Many people don't realise that genetics was a real subject, a really important subject in the early 20th century. Many think of the findings of Watson and Crick with the help of Franklin in 1953, but there was many more things going on earlier in that century. And this means we get to what we call the Hardy-Weinberg Principle. So it was the first person to really come up with this idea was Wilhelm Weinberg, who developed the principle of genetic equilibrium independently of the British mathematician Godfrey Hardy. This exposition was delivered in 1908, and this was 6 months before Hardy's paper was published in English. So his lecture, Hardy's lecture, was later printed that year in the Society's yearbook. Weinberg's contributions, they were relatively unrecognized for many years, and obviously if you study history you'll realize why. Obviously in the early 20th century there was not much love lost between German and English populations at that time. But during kind of the mid-40s there was kind of a realization that both Hardy and Wilhelm Weinberg's exposition came together, and really they came up with the same thing. So the concept of genetic equilibrium is now known today as the Hardy-Weinberg Principle. Now Hardy really he put a more comprehensive idea together, but they both have been recognized for their discoveries. The principle is greatly related to the ideas of evolution, which is generally defined as being the sum total of the genetically inherited changes in the individuals who are the members of a population's gene pool. It's clear the effects of evolution are felt by individuals, but it is the population as a whole that actually evolves. Evolution is simply a change in the frequencies of alleles in the gene pool of a population. So, for example, dominant and recessive alleles. This definition was developed largely as a result of, as I said, between the independent work of Hardy and Weinberg. And through mathematical modelling based on probability, they concluded in 1908 that gene pool frequencies are inherently stable, but that evolution should be expected in all populations virtually all of the time. You will need to learn these key terms. You've come across these key terms before, so definitely population, gene pool, and allele frequency, which you would have studied in the previous section of work. So the frequency of alleles, genotypes and phenotypes in a population can be calculated using the Hardy-Weinberg equation, where P is the frequency of one allele, usually the dominant allele, and Q is the frequency of the other allele, which is usually recessive of the gene. Now, to remember that in an exam is quite difficult. I think that's quite difficult to remember what the P and the Q is, and you can make mistakes really easily. So I've got a really good method of remembering this, particularly in terms of talking about alleles of the gene, so homozygous, heterozygous alleles. Here we have a Punnett square diagram, and I've got two heterozygous parents with big B, little b alleles, and we know that the dominant allele is P. and we know that the recessive allele is Q. So therefore, as you can see, the first example here, so there's dominant homozygous alleles, so that will be P squared. The Q squared is the recessive alleles there, and finally we have one PQ, which is the dominant and recessive allele, and the other PQ, making 2 PQ. Now, I think that's a really good way of remembering it, and particularly when you tackle an exam question and maybe you can't remember, excuse the pun, your P's and Q's. As said before, the Hardy-Weinberg Principle is a mathematical model that is predicting the frequency of alleles in a population that won't change from one generation to the next. Therefore, it's making a lot of assumptions related to the population. This includes that there is no selection of particular alleles in the population. There's no mutation. No migration, meaning that the populations are isolated. Particularly at number 4, you need a large population with a large frequency of alleles. And fifthly, you're assuming that there is random mating, so all possible genotypes can breed with all others. Thank you to Amoeba Sisters for this slide. So let's go on now, and I'm going to go through a specific example. So the example we're going to use here is the coat colour in cats. So we're going to look at the genetics regards black coat in cats and white coat in cats, and that cat doesn't look very happy. So the first thing says it's the population of 1,000 cats, 840 are black and 160 are white cats. So we need to work out the frequencies of the black and white phenotypes. So what you do is you take the number of cats and divide it by the total number of cats. So as you can see, for black cats, that's going to be 2 genotypes. You're going to have homozygous dominant and heterozygous dominant. So it's 840 divided by 1,000, which gives you 0.84. And the recessive homozygous, little b, will give 160 divided by 1,000. Which gives 0.16. So as we know, each cat has 2 alleles for coat colour, so they've got a dominant and recessive allele. So this means they can have 3 genotypes. So we don't know what the frequency of each genotype is in the population, but we do know that the sum of the 2 allele frequencies, the dominant and recessive, must add up to 1. So by definition, because there are only 2 alleles. So mathematically, P is the dominant allele B, big B, and Q is the frequency of the recessive allele lowercase b. Then P plus Q equals 1, and that's quite important to remember when you do the example. So if we go back to the Punnett square, you can see that gives us the frequency of the different genotypes in the population. And we know which frequencies relate to the equation. So to answer this question, and with any Hardy-Weinberg question in your A-level biology course, you need to look at the evidence that they give you in the question. They'll always give you a clue related to your P or your Q. Okay, so if we go back to the phenotypic frequency for black, which is 0.84, and for white is 0.16. Now we know the white cats, they are homozygous recessive, so the frequency of the genotype the genotype that's recessive homozygous is 0.16. Now if you look in your Punnett square, you'll know that that genotype is Q squared. So Q squared equals 0.16, and that is your gateway into answering the question. To find Q, all you do is you do the square root of Q squared. So if you square root it, you'll get 0.4. So Q equals 0.4. You then, by deduction, you can work out that P equals 1 minus Q. So that is 1 minus 0.4. So that P is 0.6. So now you've got P, Q, and Q squared. Now we have this information, we can calculate the genotypic frequencies. So here we go. So the first one, frequency of the homozygous dominant, which is P squared, so it's 0.6 squared because 0.6 equals P, and that gives you 0.36. You then can work out, because you know the P and the Q, you can now work out 2PQ, which is the frequency of your heterozygous genotypes. So it's 2 times 0.6 times 0.4, which gives you 0.48. You already know 0.16. Now at this stage, you should be able to know whether you're on the right lines, because all of those should add up to 1. Because we know P plus Q equals 1. And we can now convert these frequencies into actual numbers in the population. So for example, we have the number of heterozygous cats, we can work that out. That is 0.48 times 1000, and that will give us 480 cats. So that tells us that in terms of homozygous dominant cats, there is 360, and as we already knew, white cats, there is 160 in the population. So that's kind of a way of thinking about the Hardy-Weinberg principle. Let's now look at an exam question. So here's an exam question. This is a typical style of Hardy-Weinberg question. They give you plenty of information, so they're telling you the allele for the red body colour is dominant, so the allele for black body colour, which is recessive. Okay, so those they're telling you the alleles. And then it says a mixture of red beetles and black beetles were kept in a container in the lab under optimal breeding conditions. So that's assuming that certain conditions are kept controlled. And then it says after 1 year there were 149 red beetles and 84 black beetles. And it tells you to use the Hardy-Weinberg equation to calculate, so you're going to have to calculate the expected percentage of heterozygous red beetles. Okay, so the heterozygous red, which is big R, little r. So firstly, I've drawn a Punnett square, and just to remind myself which alleles are related to the equation. So P is the dominant allele and Q is the recessive allele. So I'm going to have to find, if I just write the equation there, so P squared plus 2PQ plus Q squared, what I want to find out is 2PQ. So let's have a look at the numbers first of all. So there's 149 red beetles plus 84. So that's telling us there are 233 beetles in total. So now we can work out the frequency of the one that we know, which is the black beetles. So those are going to be homozygous recessive. So basically that equals, I know from looking at my Punnett square, that's Q squared. So that will equal 84 divided by 233. And that gives me 0.36. So therefore Q equals the square root of 0.36. So that will give me 0.6. So if Q is 0.6, I know that P equals 0.4. because P plus Q equals 1. I can then work out 2PQ, so 2 times 0.6 times 0.4. Because I'm running out of space, I'm going to go back up here. It's 0.48. Now, is that the answer? Well, no, it isn't, because if you remember, it wants you to work out the percentage, which is dead easy, times it by 100, and you will get 48%. There we go. This next example, as you can see, is about sea otters. Okay, and it tells you dominant allele T, and you've got dominant allele T, and the other allele recessive does not produce a functional enzyme, and then it tells you the allele frequency for the recessive allele T was found to be 0.2. And it says use the Hardy-Weinberg equation to calculate the percentage of homozygous recessive sea otters in the population. Well, okay, homozygous recessive is TT. Now, the thing is, we already know T equals 0.2. So to find the recessive, homozygous recessive, you just times it by 0.2, and that gives you 0.04, and times it by 100, and that gives you 4%. In some questions they will give you some data. In this case they're showing the frequency of some alleles in the population of cats in 3 cities. So you've got the cities and you've got then the frequency of alleles. Now the first question tells you that white cats are deaf, and it says would the Hardy-Weinberg principle hold true for white cats? Okay, well the first thing we need to think about is the conditions required for the equation, for the principle, sorry. And if you think about it, if white cats are deaf, are they likely to survive? They've actually got selection pressure against them, okay, so therefore they won't survive and they will not pass on the allele, and that therefore will reduce that frequency of that allele. And then the next question says, what is the evidence from the table that non-agouti and blotched are alleles of different genes? So looking at the table, if you look at the Paris and London frequencies for non-agouti and blotched, so particularly, for example, Paris here, these add up to more than 1, so it equals 1.49. So the frequencies in Paris or London, you can mention London as well, they add up to more than 1. Okay, so this is why they are alleles of different genes. Okay, and then the final part of this question talks about hair length in cats, and it tells you it's a single gene with 2 alleles, so it's going to be Hardy-Weinberg. So the allele for long hair, the long hair is actually recessive in this case, and the allele for short hair is dominant. And it does say, most importantly, use the information in the table and the Hardy-Weinberg equation to estimate the percentage of cats in London that are heterozygous, so London, that are heterozygous for hair length. You have to show you working. Okay, so it's important to use the information they give you, and the first thing is, notice that it says the frequency of the allele. Okay, alright, so it doesn't say alleles, it says allele. So this figure here for long haired is the recessive allele. So in fact, that is Q. So Q is 0.33. By deducing that P plus Q equals 1, we know that P will equal 0.67. So therefore, 2PQ, you just take 2 times 0.33 times 0.67. Do that on your calculator and you will find the answer of 0.4 4, and it says the percentage, so it's times 100, and that will give you 44%. So this is a really good example of reading the question, okay? All right, it says the frequency of the allele, so it's not Q squared, this is Q. All right, so that's the difficulty with this question. I hope you've found this Hardy-Weinberg or Weinberg masterclass useful. I'm sure the White Cat has found it really, really useful. So there you go. Give him a big smile. I hope you found it. So if you found it useful, please do subscribe to DrBiology, and there'll be more A-level masterclasses coming up soon.